Hey everyone, in this video we are going to look about the steps that are required to prepare and stain a specimen for viewing under microscope. So we'll be discussing about fixation. So why is it done? It is done to increase the visibility of the microorganism under the microscope, and it helps to accentuate the specific morphological features of these organisms and helps preserve them for further studies without damaging the cells. So fixation. So it is a process by which internal and external structure of specimens are preserved and fixed in position. It inactivates the enzymes and toughens cell structures so they do not change during staining and observation under microscope. Alright, so here is one of the method of fixation, which is heat fixation. So as you can see in the figure here, there is a glass slide onto which, using an inoculation loop, we are transferring a drop of the culture onto a drop of water and mixing it, and then we are actually moving the slide under the flame for a few seconds. So this method is used to observe the bacteria and archaea mainly, and in this a film of cells or smear is gently heated as the slide is passed through the flame. So it actually helps preserve the overall morphology, inactivates enzymes, and destroys proteins, part of subcellular structures. Alright, so the next type of fixation is chemical fixation. It is a technique used to fix a specimen, like biological tissues, with chemicals to prevent autolysis by the action of enzymes and deformation of morphologies during specimen preparation. So example is fixation of a specimen for transmission electron microscopy. So in this chemical fixation, what happens is that it can help protect the fine cellular substructure and morphology of larger, more delicate organisms, instead of just simply killing and completely denaturing it. The chemicals penetrate the cells and react with cellular components like proteins and lipids, making them inactive, insoluble, and immobile. So here you can see a transmission electron microscope, and as we said, the chemical fixation is done for some specimens which have to be observed under the Temp. So for the Temp observation of a biological specimen, the specimen must be prepared by removing water to preserve its morphologies under vacuum in the microscope column, and by thinning the specimen to transmit electron beams. So for all these requirements, sometimes this dehydration and resin embedding and thinning the sectioning, thin sectioning, etc., can cause deformation of fine structures of specimen. So as you can see here in this figure, they are showing some of the steps under which the cell culture undergoes chemical fixation and cryofixation, etc. And it is sectioned and then imaged under the Temp microscope. Alright. So as you can see, here are some of the very clear images which are observed using a transmission electron microscope. Alright, so as we said, chemical fixation thus preserves the morphologies and physical properties at the living state of the specimen as much as possible. And this technique prevents the autolysis and deformation by cross-linking the proteins or lipids of biological materials using chemicals. Now, the common fixative mixtures may contain ethanol, acetic acid, mercury chloride, formaldehyde, and glyceraldehyde. Alright, in the next part of the video, we'll be learning more about the dyes and symbol staining and differential staining, which are an essential step to view the microorganism under microscope. Thank you.